We have a little short lecture on fungi, and then we'll start talking about viruses, and we'll go over some general characteristics of viruses first, and then we will start to focus on the bacterial viruses. So fungi, we had a little lab where we looked at some fungi. I told you some things about the fungi, but they are not plants, although originally they were placed into the plant kingdom because once upon a time there were only two kingdoms, plants and animals, so they stuck everything that wasn't an animal into the plant kingdom. But fungi are not plants. They do not have the vascular tissues that plants have. And you may recall plants have xylem and phloem, which is how they conduct water and food throughout the plant. No xylem or phloem in fungi. Although some fungi like penicillium actually are green, the green pigment is not chlorophyll, so there's no photosynthesis in fungi. They are eukaryotes, and they can either be aerobic or facultative anaerobes. When we classify the fungi, we classify the phylum based on the sexual phase of a fungus. Remember I said that it may not be possible to actually observe the sexual phase, or it may take a long time before scientists finally do observe the sexual phase. But most fungi eventually do get classified. So we're going to look at some of the major phyla, not all of them, but some of them. Our first phylum is Zygomycota, where we have the Zygomycetes. Remember I said when you see the word mycete, you should think fungi. Even though the actinomycetes are prokaryotes, they still hold on to that name. But normally mycete means fungus. So the Zygomycetes, we have two examples, Rhizopus and Mucor, but the images we're looking at are both of Rhizopus. On the left, we have an agar plate with the Rhizopus growing on it. You see it's mostly sort of white or colorless fuzz, and you see the areas with all the black dots. Those are sporangia, so the aerial hyphae that are reproductive. And the sporangia are full of spores. And we're looking at asexual reproduction here. But as we mentioned, fungi do get together and exchange genes and reproduce sexually. On the right, we have Rhizopus zygotes. So remember, the zygote is a diploid structure. It's produced when two mating strands come together and they exchange genes and they form a zygote. The zygote will produce spores, and the spores will be haploid because spores are always haploid. So fungal spores are always haploid. Whether they're produced through mitosis, haploid to haploid, or through meiosis, diploid to haploid, spores are always haploid because most fungi exist as haploid organisms. And what did we say was the benefit of sexual reproduction? What's the benefit? Right, variation, right, to mix up the genes. And here we see when we grow Rhizopus on an agar plate, like what we see here, we have to tape the plate shut. And the reason why we have to tape that plate shut is because Rhizopus grows so vigorously, it will literally push the plate or push the lid off the plate. And then it will spread the spores all over your workspace. So Rhizopus does grow very thick, very dense, and it will push the lid off a Petri plate. So we have to tape that shut. So all these filaments, those are all hyphae. Our next phylum, Ascomycota, where we have the Ascomycetes. And remember we looked at a slide of Aspergillus, and we saw it was very pretty, looked kind of like a flower, but I said it was a slice of a conidiaphore. So here we see the conidiaphore. Notice that it's covered with spores. So every conidiaphore has hundreds of spores on it. And the fungal colony produces a lot of conidiophores. So fungi make thousands or hundreds of thousands or millions of spores. And when you take a slice of this and stain it, it looks very pretty, like a flower. And so Aspergillus is the main type of Ascomycete that we talk about. There are different color varieties, as we see in this agar plate here. We have some Aspergillus that's basically kind of white or non-pigmented. We have golden yellow, we have brown, and we have dark brown. So you have different color varieties. And when it comes to fungi, when they start to develop color, that's an indication that they're reproducing and making spores. Now, why are we concerned about Aspergillus? Because Aspergillus produces aflatoxins. Aflatoxins are carcinogens. What's a carcinogen?
Something that can cause cancer?
Right, so something that causes cancer. And so there are actually certain food products that are tested for aflatoxin. And the most common one is peanut butter. And the reason why is that when the peanuts are harvested, if they are not allowed to dry properly, they can get damp and moldy. And if one of those molds is Aspergillus, it will contaminate the peanuts with aflatoxin. And so they do test certain products like peanut butter for aflatoxins to make sure they don't have any. Most people are familiar with the Basidiomycota, not by that name, but the Basidiomycetes are the fleshy fungi, like mushrooms and toadstools, or shelf fungi like you see growing here on dead and dying wood. And so one of the main functions or purposes of fungi is to decompose, dead matter, especially dead plant matter. So you see these shelf fungi growing on either dead wood or trees that are sick. And some of the shelf fungi can be very large, like almost a foot across, growing off a tree. So they can be very, very large. Most people are familiar with mushrooms. Does anybody like mushrooms? Who likes mushrooms? Clarice loves them. I think they're disgusting. I cannot stand mushrooms. It's not the taste, it's the texture that grosses me out. I just can't stand that kind of, chewy, weird kind of rubbery texture. Yeah, I just don't like that at all. Now, most mushrooms are actually poisonous, right? Only certain mushrooms are edible. So obviously, if you buy them from the store, you're getting the edible kind. If you like to go out to the woods and collect your own, you really need to know for sure that you're collecting the right kind of mushroom. Otherwise, that will be the last mushroom you will ever eat. Now, the mushroom has a stalk and a cap, and the cap is actually the diploid structure. And if you look underneath the cap, you see gills, which again looks kind of nasty. If you want to do an experiment, if you have some fresh mushrooms, cut the cap off and put it on a piece of white paper, leave it there overnight, and you'll usually see spores that have dropped down from the mushroom cap. So the cap is a diploid structure that's producing the spores. And if you take the stalk and you actually dissect it apart, you'll see that it's actually made up of filaments. It's made up of the hyphae. So the hyphae are versatile structures that a fungus can use to create more elaborate structures. If you cannot find the sexual phase for a fungus, then you put it into Deuteromycota, also known as the fungi imperfecti, because there's an asexual phase. But like I said, usually at some point, scientists eventually discover the sexual phase and they can, put it into the proper phylum. But if it's all asexual and they just cannot find a sexual phase, they put it into Deuteromycota. Now the last phylum, I just want to mention the Oomycota because they are no longer in the fungus kingdom. Oomycota have been reclassified into Protista, which, as you recall, the kingdom Protista is a catch-all kingdom. You have all different kinds of organisms that don't fit into the plant, animal, or fungus kingdom, so they stick them into Protista. So the Oomycota, which includes things like Saprolegnia, which is a water mold, and we actually used to culture this and use it in our microbiology lab a long, long time ago. It's no longer considered to be a fungus. So it just didn't have enough of the characteristics, so they decided to move it into Protista. That was a while ago. Things might have changed by now. That's one of the problems with classification, is that it is constantly changing. But we classify fungi based on the sexual phase and then put it into the proper phylum. Now, when it comes to the kinds of fungi that we're most concerned about in this course, those are yeasts and molds. As I mentioned last time, the molds are fuzzy, they're filamentous, and the yeasts are slimy. So in a filamentous mold, those filaments are the hyphae or hyphae. And if the hyphae are well organized into a thick structure, that's called a mycelium. And this part of the fungus does require oxygen, so it's aerobic. So molds typically are aerobic organisms. They must have oxygen. But yeasts, which look like large cocci, are single-celled microorganisms. They form colonies like bacteria, although yeast colonies tend to be a lot more raised than a bacterial colony. And yeasts are facultative anaerobes. So they don't need oxygen to grow, but they do grow better with oxygen. Now, there are some fungi that exist only as a yeast, some fungi that exist only as a mold, and there are certain fungi that can actually be both, a yeast or a mold. So we call these dimorphic fungi. So they exhibit dimorphism, two shapes. And what determines whether or not they're a yeast or a mold are environmental conditions like temperature, nutrient availability, CO2 in the atmosphere. But if you have a dimorphic fungus that is a mold at room temperature, so RT means room temperature, if it's a mold at room temperature, it turns into a yeast at body temperature. So there are certain fungal pathogens which at room temperature, it's a mold, but if it somehow gets in you to infect you, it turns into a yeast. If we examine the filaments, the hyphae microscopically, we see two major types, septate and aseptate. Septate hyphae have divisions in between cells. Aseptate hyphae have no divisions, but you would need a microscope to actually see this. If you take the hyphae and you pack them tightly together, you can form a structure like the stalk of a mushroom, which technically is called a thallus. And so, as I said, the hyphae are versatile structures that can be assembled to make much larger, much tougher structures than the actual hyphae themselves. So back to molds. Most of the hyphae that you see, especially when a mold colony is young, are the vegetative hyphae. And they actually are used for feeding the fungus. So they're growing on a substrate, and the ends of the hyphae release digestive enzymes. And then nutrients can be absorbed through the hyphae, and that feeds the fungus. And hyphae typically, the vegetative hyphae are typically colorless. But as they mature, they start to produce these aerial hyphae, these reproductive hyphae that contain spores. And that's usually when the fungal colony begins to get colorful, is when it's making these spores. And some fungal colonies are actually surprisingly, pretty because they're very colorful. Now, yeasts are single-celled organisms, so they're not filamentous. They're facultative anaerobes, and they primarily reproduce by budding, which is an asexual form of reproduction. So when it comes to reproduction and the formation of spores, remember spores are always haploid. Whether they're produced from a haploid organism through mitosis or from a diploid structure like a zygote through meiosis, spores are always haploid because most fungi exist as haploid organisms. And as I mentioned, the fungi produce a lot of spores. A fungal colony will have thousands or maybe even millions of spores, and they're very, very light. So just a slight amount of air movement and the spores will float away, because the purpose of fungal spores is reproduction, so they make a lot of them. Now, the spores, a fungal spore has some resistance to the environment, but they're nowhere near as tough as a bacterial spore is, because the fungi are simply trying to reproduce. And so fungal spores are everywhere. They're floating through the air as I speak right now. We're all inhaling fungal spores. They land on your food, you eat them. So they're always there, right? They're all around. If they happen to land on some of your food and it's something that's, you're leaving, for a long period of time without touching it, either on the counter or in the refrigerator, over time that fungal spore will germinate and may produce a colony of fungus. As I mentioned before, if there's little green spots on your bread, just pick them off because they're only on the surface. If it's a green colony on your block of cheese, just cut it off. The rest of the cheese is fine. It's only growing on the surface, but mold spores are ever present. They're always there. So again, the main point about reproduction and the formation of spores is that they are always haploid. Now, when it comes to yeast, we already said that yeasts can simply divide in two. It's an unequal cell division, but they divide in two. And so it's an asexual reproduction. And as the division is occurring, because it's not an equal division, the new cell that's forming is smaller than the original parent cell. And so we call that new cell a bud. Technically, it's a blastospore, but we'll just call it a bud. Eventually, the bud is going to reach full size. But during the initial formation, the unequal cell division, the original cell remains large, and then a smaller cell buds from it. So this is all asexual. Sometimes yeast do get together and exchange genes in sexual reproduction and form spores. Technically, those spores are called ascospores. Here we see a stain slide. Remember, the yeast cells are blue. Inside are the dark purple, almost black spores, which are found inside those yeast cells. And we said that yeast reproduce sexually when they're stressed. So when they sense the environment is deteriorating, maybe it's drying up, maybe they're running out of food, or it's becoming polluted and toxic. And so the yeast cells will get together, exchange genes, and form the spores. And the idea is by mixing up the genes, you increase genetic diversity. And so the offspring, hopefully, will be better adapted to survive in that environment. But if the environment's wonderful, you don't need that diversity. You can just bud, right? So you can just clone yourself because there's no need, right, for genetic diversity if everything is wonderful. But it's when things are not wonderful that yeasts and other fungi get together and exchange genes, right? They need to mix up those genes and create that variation. When we grow fungi, we use special agar plates. So the agar that's used is saborodextrose agar. It's an agar that is low in pH, so it's acidic, which can inhibit bacteria. It's also high in sugar, so it's hypertonic, which also can inhibit bacteria. And so does anyone know what we call that kind of medium that allows some microorganisms to grow while inhibiting others? What do we call that kind of medium, which allows some microorganisms to grow but inhibits others?
Selective
Yep, a
medium?
selective medium. So we need to use a selective medium because very often bacteria will be contaminating our cultures or the agar plates. And so we need a way to stop the bacteria from growing. And so we use a lower pH and higher sugar, which the fungi either like or tolerate very well, but the bacteria cannot. So as I already mentioned, the primary role of fungi in the environment is as a decomposer, technically a saprophyte, an organism that eats dead things. So when trees fall down, when plants die, molds grow on them, fungi grow on them like shelf fungi, and the whole purpose is to help to break down that dead plant and recycle nutrients back into the environment. Fungi can also be parasites that can be pathogens to plants and animals. And when it comes to plants, if your parents do any gardening, they can probably tell you a common fungal infection on plants is something called powdery mildew. It's just a fungus that grows on the leaves. Roses are notorious for getting other kinds of fungal diseases, but there are different kinds of fungal infections that affect plants. Sometimes you can spray the plant to help protect them or save them. Other times the plants get sick, maybe all the leaves drop off. Sometimes the plants die, but other times the plants, will survive this. But it's not a good thing. When it comes to animals, most of the time, as long as you're healthy, have a healthy immune system, you will not get a serious fungal infection. Often we talk about topical skin infections like athlete's foot or jock itch, which are easily treatable. But as long as your immune system's healthy, you're not likely to ever get a serious fungal infection because they can and will be very serious if you actually get one internally. And if you look through any microbiology textbook, through the disease chapters and the chapter on fungal diseases, you'll see how nasty they can be. Remember, they're harder to treat because they're eukaryotes. There's one example of a mutualistic relationship. Remember, a mutualism, both organisms benefit. In this case, we're talking about a lichen, or if you're British, a lichen. And this is a combination of a fungus and a cyanobacterium. Remember, cyanobacteria are blue-green algae. So if you've ever, been hiking through the woods or something, maybe you've seen on the bare rock, it's this kind of flat, light green patch of something growing on it here. Have you guys ever seen that on bare rocks, these flat, greenish patches that may be growing on the rock? Anybody ever seen those? Well, no, it's flat. So it's not fuzzy or hairy, it's just flat. And so those are actually lichens. And so, but it's growing on bare rock, which is a harsh environment to grow on, right? Because there's no soil, right? The only water it gets is when the rock gets wet from rain. And so this fungus and cyanobacterium would not be able to survive by themselves, but together they can survive because the fungus will absorb the moisture when it rains. And the cyanobacterium is a type of algae, so it can photosynthesize, right, to make ATP and sugar, right, to feed the fungus. And so they live together in very harsh environments. As far as benefits go, it's mostly in the food industry. Yeasts in particular make bread rise. Remember we talked about fermentation. Yeast produce ethyl alcohol and carbon dioxide gas. And so the gas bubbles get trapped in the dough and cause it to rise. And that's what makes your breads light and fluffy. We can also use yeast to make beer and wine and champagne, if you like that kind of stuff. And then some people actually eat these nasty fungi directly, like mushrooms. Or if you like blue cheese salad dressing, the chunks of blue are mold. And so many cheeses do contain mold. So there's all kinds of nasty fungi that different kinds of people like to eat. And they are harmful, right? Fungi can cause diseases in plants and animals. When it comes to food crops, then that can be potentially serious, because if you ever looked up the Irish potato famine, a long time ago, the Irish, lots and lots of them starved to death because their primary source of food was potatoes. It got infected with a fungus, and it basically killed most of the potato crops, so they had nothing else to eat. Right, so fungi can cause some serious damage to crops. As I said, for humans, as long as you're healthy, have a healthy immune system, you're not likely to ever suffer from a serious fungal infection. But if you see the word mycosis attached to something, that indicates a fungal disease, like blastomycosis or coccidiomycosis. That tells you it's a fungal infection and a fungal disease. So any questions on fungi?
I have a question. So, if there's mold on cheese or bread, does that mean it's decomposing it? Or it just fell on there and it's growing?
Right, the spores just landed on there and they germinated and they're growing and eating it, right? Because your bread and cheese are not living things, right? So it's like dead matter that needs to be decomposed.
So they decompose only living things?
No, dead things. And your bread and cheese are not alive, so.
Right, but I'm saying, how can you tell the difference if it's the bread is really bad or the cheese is really bad, or if the spores just fell on it?
Oh, because the molds are only going to be growing on the surface, right? Because the molds need oxygen, so they're not going to be growing inside your block of cheese. So just on the surface. So if you cut it off, the rest of the cheese is fine.
Okay, thank you.
I mean, if it's completely covered in green fuzz, yeah, just throw it away. But if it just has some green spots on it, just cut those off.
Okay, thanks.
Okay, so now we're going to change gears and start talking about viruses. And I'm going to talk a little bit longer today because lab is short. So some general characteristics of viruses. Viruses do contain nucleic acid, but it's one or the other, not both. So it's either DNA or RNA, but not both of them. And that nucleic acid is surrounded by a protein coat. Does anybody know what the fancy name for the protein coat is? Anybody know the name for the protein coat? Capsid. All right, so the protein coat is called a capsid. And then some viruses, especially certain animal viruses, are covered with a lipid membrane envelope. Some viruses have protein spikes that stick out from them. And it's the spikes that help those viruses to attach to host cells. And viruses do tend to be very host specific, infecting only one or maybe a few types of hosts. And within that host, they tend to be very cell specific, only infecting one type of cell. So like respiratory viruses, they only infect respiratory epithelial cells. not heart cells, not liver cells, not bone cells, just respiratory epithelial cells. And that's determined by the cell surface receptors that are on the surface of those cells. And so the viruses have evolved to use just those particular cell surface receptors to attach and infect a cell. Now, there are some viruses that I call promiscuous viruses because they can infect a wide variety of hosts, like flu viruses. All viruses are obligate intracellular parasites. So that means they must live inside of a host cell. Every living cellular organism is a host for some kind of virus. Invertebrates, vertebrates, plants, protists, fungi, bacteria, even the archaea have archaeviruses that infect them. So a hyperthermophile living at 100 degrees Celsius, there's some virus out there that can infect that archaea. And so what does that suggest about how long viruses have been around?
Forever.
Right, for a very long time, possibly from the very beginning. And in fact, scientists don't know which came first. Some theories state that viruses actually evolved first, and then they led to the evolution of cells. Other theories suggest that viruses may be degenerate cells, cells that just lost so much stuff, they lost the ability to reproduce independently. Now, there are some viruses called giant viruses, or giruses for short, and these are enormous viruses. They are as big or bigger than some bacterial cells. We can actually see them using the microscopes we have in lab. They're that big. And when scientists first saw them, they just assumed they were bacteria, right? Because they were so big, right? They could see them in an ordinary microscope. But when they looked more closely, they realized there's no cytoplasm, there's no organelles, there's no nucleus, right? There's no nothing, and therefore they are viruses. But they're huge, they're enormous, and they actually have genes that we only find in cellular organisms. And so the giruses, could represent degenerate cells, cells that just lost so much stuff they were no longer cells and could not live independently. So we really don't know where viruses came from, but they probably have been around since the beginning, either at the very beginning or shortly thereafter, because every living thing has some kind of virus that can infect it. And our genomes are full of the remnants of ancient viruses. Viruses actually shaped our evolution. Your genes, your chromosomes, you have a lot of ancient viral DNA in your genome. So viruses have been around for a long time. When it comes to size, remember viruses, most of them are very small, so we use nanometers to measure them. Most bacteria, we use micrometers, but remember there are extremely tiny bacteria, the nanobacteria, which you need nanometers to measure them because they're so tiny. All viruses are intracellular parasites. Most bacteria are not. Right, so most bacterial pathogens are on the outside of your cells. But there are some exceptions like the rickettsias and chlamydias, which are very small and live inside of your cells. Viruses do not reproduce by dividing in two. They use cells as factories to mass produce up to hundreds of new virus particles. Of course, bacteria simply divide in two. We already said that viruses only have one type of nucleic acid, but all cells have both RNA and DNA. Viruses do not make their own ATP. There's no electron transport chain, no ATP synthase in a virus. Viruses don't make any proteins. There's no translation, no protein synthesis. So there's no ribosomes in viruses. When it comes to antibiotics, We do not currently use antibiotics to treat viral infections. That does not mean that certain antibiotics do not have antiviral activity, because some do. And I mentioned this during the antimicrobial drug lecture. And an antibiotic like azithromycin, which you may know as a Z-Pak, which you've probably taken, it turns out that azithromycin actually stimulates the immune system to attack viruses. So indirectly, that antibiotic has antiviral activity by stimulating your immune system to attack viruses. So it's possible in the future we may use antibiotics along with other drugs to help treat certain viral infections. So we do not use antibiotics to treat viral infections, but some antibiotics actually do have antiviral activity. And when it comes to filtration, remember we said you can filter sterilize things. Well, filters mostly just remove bacteria, not viruses, because they're so small, they get through the pores of the filter. So you may need to use an additional treatment like UV light or something in order to make sure there's no viruses present. With bacteria, remember the mycoplasmas, no cell wall, so they're not ridge cells. Mycoplasmas can often get through the pores of standard size filters because they're very flexible. They're not rigid. They can work their way through. When it comes to size comparison, on the right here, we just have one little piece of a red blood cell. Remember, this red blood cell would be huge, right? Really, really big. And you can fit E. coli into a red blood cell and many, many viruses into E. coli. So most viruses are teeny tiny. Some of them are large like Ebola, but you still need an electron microscope to see it. And something really tiny like a rhinovirus or this little tiny bacteriophage here. So viruses can be extremely small. Now there's a really cool animation I'll show you. I know the link is on my web page. I don't remember if I moved it over to black or not, but it's a nice little animation. So we all seeing the coffee bean and a grain of rice.
Yeah,
So,
we can see it.
yeah, so things that you're familiar with in terms of size, a nice, easy to read 12-point font. And so we're going to zoom in and get closer and closer. A grain of salt is 0.5 millimeter, so most of us can see grains of salt still, although we may need glasses to help us. The amoeba, again, around 500 micrometers, which you can still see if your eyes are good. But we're getting smaller and smaller. A skin cell, if you guys took one, well, if you took 150 back when we were meeting face to face, you may have done a cheek smear and actually seen your cheek cells. They're about 30 micrometers in size approximately. Here we have an egg, right? That enormous egg, and yet a little bitty tiny sperm. How come the egg is so big and the sperm is so small? Any idea why the size difference? It's a little bitty sperm, but it's also powerful, right? The sperm is just a chromosome delivery system. It's just carrying dad's chromosomes to the egg. The egg is the actual cell and provides everything needed to make a cell. All the cytoplasms in the egg, all the mitochondria, all the Golgi, all the organelles, they're all in the egg. All the sperm does is contribute chromosomes from dad. So yes, men really are just sperm donors. Zoom in closer and closer, a red blood cell, an X chromosome, and a yeast cell. The X chromosome is bigger than a yeast cell. So the cell is a living organism. And this chromosome is bigger than that living organism. It's huge. E. coli and a mitochondrion. How come the E. coli and the mitochondrion look so similar? A similar size, similar shape. Both have a circular chromosome. Why are they so similar?
Is it because of the theory that we spoke about?
Yep, the endosymbiotic theory, right? That mitochondria and chloroplasts came from prokaryotic cells that got swallowed by another cell. And that's why they're so similar. Closer, we have some viruses, teeny tiny viruses, a bacteriophage. We'll see some of that later on. Notice rhinoviruses. Rhinoviruses are very common causes of colds. They're very common. And a rhinovirus is the same size as a ribosome, right? So the little machine that actually translates and makes our proteins is the same size as a virus that causes colds. Zoom closer and closer, hemoglobin and transfer RNA, and antibody, phospholipids, glucose, and then a single carbon atom. So it's a nice little animation for a sense of scale. So you should check that out. So let's get back to where we were. If I can get this to scroll, it's not scrolling. So when it comes to shapes for viruses, there are a few viruses that are helical. And so the capsid is helical, and the capsid is made of regular repeating protein subunits called capsomeres. So the capsomeres assemble to form the capsid. In this image here, you're looking at Ebola, which looks like a snake in the electron microscope. So if you're a researcher and you're looking at some tissue, you see this, you should be very afraid. But most viruses are variations of a polyhedron. So very geometric. You have polyhedrons, icosahedrons, so regular geometric shapes are what we typically find for most viral capsids. And again, all the protein subunits are the capsomeres, which make up the capsid, and that surrounds the DNA or RNA. In this image, we see protein spikes, which virus is used for attachment to host cells. Here we see a lipid membrane surrounding this virus. So this is typically only seen in animal viruses. And the membrane comes from the host cell. So these viruses bud from the host cell, stealing host cell membrane, which eventually kills that host cell. So here we see a complex virus, which happens to be a bacteriophage. Remember, the word phagy means to eat, so a bacteriophage is a virus that eats bacteria. Not literally, it just uses the bacterial cell as a factory to make more viruses, and in the process it kills that bacterial cell. And so we have the regular polyhedron head here, and then we have all these additional structures, the sheath and tail fibers. And this is what makes this virus complex, all these additional structures. Here we see three phages which have landed on a bacterial cell. And as we'll learn more next week, the bacteriophage actually kind of drill a hole through the cell wall using the enzyme lysozyme. And then the sheath contracts and squirts the DNA inside. And here we see this blue strand of DNA being squirted inside this bacterial cell. but we'll get more into that next week. When it comes to naming viruses, remember the nomenclature for viruses is really artificial because viruses are not living organisms, interbreed. You remember a viral species is just a group of viruses which have the same kind of genetic information, the same kind of genes, right, the same genome, and they share a common ecological niche, which means they infect the same kinds of hosts. And there are different strains and subspecies of viruses. But family names end in viridae, or viridae, and the genus names end in virus. So some examples, the herpes viridae family, and the genus herpes virus, and the species human herpes virus. And as we'll learn next week, there are 8 known human herpes viruses, and you'll learn about all of them. And they're designated as human herpes virus 1, 2, 3 through 8. We still use the old names for the herpes viruses, but the newer designation is human herpes virus. But the genus is herpes virus, and the species is human herpes virus, 1, 2, whatever. So it's not your typical genus and species. The Retroviridae family, we have the lentivirus genus, and the species, HIV, is an example. So in genus and species, it doesn't work the same way with viruses. When it comes to growing viruses, this presents a problem. Viruses are not living things, or not cellular organisms. You can only grow them inside of a host. For bacterial viruses, it's not too difficult. You use bacteria as your host for the viruses. And this is actually an experiment that we're going to do, quote unquote, so we're going to talk about this, but you mix your virus and your bacteria together and spread that mixture on the surface of an agar plate. As the bacteria grow and reproduce, they completely cover the entire surface of the agar with bacteria. As the viruses reproduce, they kill the bacteria and leave holes in the growth. We call those holes plaques. And so wherever you see these dark spots, those are clear zones, plaques, which are full of virus. And we're actually going to count the plaques because this experiment is another serial dilution. So we'll go over serial dilutions again. We'll count plaques, multiply by the dilution factor, but this time we get PFUs per liter, not CFUs. A PFU is a plaque forming unit. And what forms a plaque is one virus particle. So it's a concentration of viruses per milliliter in the original sample. So it works the exact same way when we did the serial dilutions for bacteria, but instead of counting colonies, we're now counting plaques. Now what's interesting about these plaques, this is what you would see after 24 hours. And it doesn't change in 36 or 48 hours. The plaques stay small. So even though the viruses are sitting there touching other bacterial cells, they will no longer infect and kill those bacterial cells. And so the question is, why not? Why don't the viruses continue to spread and wipe out every bacterial cell on that plate? And the reason has to do with the bacterial growth curve. So under ideal conditions, the bacteria enter stationary phase relatively quickly, soon to be followed by death. If you're a virus, what kind of cell do you want to infect? A stationary phase cell, right, just sitting there hanging on? Or do you want a log phase cell, which is very metabolically active and actively dividing? And so, of course, you want a log phase cell. Log phase is short, right? So the viruses only infect during log phase. After that, the viruses just sit there and wait for better hosts to come along. So does that make sense?
Yeah, but they're still living bacteria during the stationary phase, so they don't even
Yep,
bother with that.
right, but they're not very active, right? They don't have very high metabolic rates. And so the viruses are just not interested, right? They want young, healthy, vigorous bacterial cells.
see. But does that only apply to bacteria? Because let's say for humans or animals, viruses will still attack the weaker people.
Oh yeah, I mean viruses attack old people as well as young people, right? But they may not reproduce very quickly, right? Warts do not grow very quickly, even though they've infected your skin cells. Warts tend to grow very slowly, right? Because your skin cells are not dividing very rapidly. When it comes to animal viruses, we have a problem. We need an animal host. So very often what are used are fertilized chicken eggs. Not the eggs you buy in the grocery store. Those have not been fertilized. They're not baby chicks. They will never become baby chicks. They're just an egg cell. But a fertilized egg has an embryo growing in it. And the embryo is experiencing rapid mitosis and cell division, so high metabolic rate, and that's what the viruses want. And so chicken eggs are commonly used to grow animal viruses. In particular, we use them to grow flu viruses to make the flu vaccines. And so often they will ask you if you are allergic to eggs. And the reason why is because the virus is grown in chicken eggs. And during the purification process where they're making the actual vaccine, may not be possible to guarantee that every egg protein has been removed from that vaccine. So if you were severely allergic to eggs, you might have a serious reaction to the vaccine, but that's rare. But they do use fertilized eggs to grow viruses. But in the past few years, they've come to realize that for the flu vaccine, growing the flu virus in eggs may actually not be the best way to do that. And then they have to find an alternative way to, to make more virus so they can make the vaccine. But growing it in the chicken eggs may not be such a good idea. It may not be quite as effective.
Yeah, I don't know the science behind it, but it doesn't sound like a good idea.
Well, it's just that the embryo is not really the natural host. And so the virus is not actually in its perfect wild type state, and so may not represent the best form of the virus to use for a vaccine, although it still works pretty well if they get lucky and actually make the vaccine for the correct strain of flu that reaches us. But we'll talk about that later and how that's a guess. And some years they guess right, and some years they guess wrong. But they may have to start growing the virus in other ways in order to make the vaccine. We can use tissue culture as a way to grow viruses, right? Because you can grow animal cells in a petri plate in a tissue culture. And one of the benefits to doing this is you can observe the effect of the viral infection upon those cells. And as we'll learn next week, that can help us to identify what kind of virus it is. But we'll get into that next week. So we got to stop here for today. And so we'll start lab at about 10 after 1. All right, any questions? Okay, then I'll shut this down to get ready for lab. So see you soon.